Um, Hello everyone. I'm really happy to welcome Rob today among us. In addition to being my friend, he is a great scientist. Rob works on the ecology and evolution of cancer. He does modeling. You will see that later, both mathematical and computational. But also he collaborates with people doing experiments and with clinicians. So he did his PhD at Oxford, and then he moved to Montpellier to work on cancer. So moving from host-parasite interactions to then cancer. This is where we met actually. And then he did a second postdoc at ETH in Zurich in Switzerland, both with Nico Berenwinkel and Hannah Coco on tumor evolution and also on evolution of cancer risk. And since 2020, he's back in the UK. He is working as a senior lecturer at the University of London. And I thought it would be very interesting for us to hear about his work, both for people interested in cancer, but also for people interested in modeling and how like theoretical approaches can be used in collaboration with more experimentalists and clinicians. So I hope you will enjoy it. And Rob, it's yours.
Thank you very much for the introduction, Mary, and thank you, Thomas, for the invitation. I had hoped to come to Bordeaux in person because I was on sabbatical in Montpellier late last year, but it didn't work out that time. But I'd love to come another time and hear more about what your institute is doing and your group in particular. So, as Mary said, I use evolutionary theory and mathematical and computational models to try to understand biological systems, particularly cancer. One of the main threads of my research is seeing whether we can use these evolutionary and mathematical methods to improve cancer treatment strategies. And that's what I'm going to focus on today. So I'm going to start, and this was a suggestion that Thomas had, which is I think is an excellent one, to start with the main messages at the beginning. So these are, if you take nothing else away from the talk, then these are the messages I'd like you to remember. The idea here is that evolutionary theory can suggest new cancer treatment strategies. So rather than developing, or as well as developing new drugs or other new treatment methods, the concept here is to use existing therapies in a smarter way, informed by what we know about the evolutionary development of tumors. And in order to understand those evolutionary dynamics and to predict how a tumor might respond to a particular strategy, we need to use mathematical modeling. And we can do that to identify the most important parameters and to predict when this strategy might be appropriate or when we might try some other strategy. And of course, working with experimentalists and clinicians is how we can parameterize those models and also design appropriate models. So integrating the modeling with the experiments from the start is what I really try to do. And I've been working with a lab in Montpellier doing that for some years now. And ultimately, at this stage, what we want to do is to inform the design of clinical trials. So I'm going to focus on two of the strategies we've been investigating. The first one, the basic concept is if we can't cure a cancer, if we think based on clinical experience that there is little prospect that the available treatments will cure a cancer, then instead what we might hope to do is to contain tumors and suppress resistance and therefore maximize survival and quality of life rather than aiming for a cure. But if we can cure, if we know that a particular treatment in a particular cancer, in a particular cohort, has a reasonable success, reasonable chance of success, then what we want to do is to maximize that cure probability. And so we're going to look at a strategy where we switch treatment sooner before the first one has failed. You can think of this as a kick it while it's down strategy. So these concepts are not my own. The hypotheses actually stemmed from Bob Gatenby at the Moffitt Cancer Center in Florida. So Gatenby is a pioneer in this field. He came up with these evolutionary strategies and the idea of using mathematical modeling to design new approaches to cancer treatment. Um, and he not only started working in this area, he put a lot of effort behind it and set up an entire department of integrated mathematical oncology at the Moffitt Cancer Center, and they followed that up with evolutionary tumor boards and all kinds of other developments. So it's the Moffitt Cancer Center, the center that's really driven these kind of approaches forward. So I'll be referencing that a fair bit. The image there comes from a 2009 essay that Gatenby wrote to set out a kind of manifesto of these kind of approaches. So these are the ongoing clinical trials of these evolutionary cancer therapies. There are already quite a few trials in progress. Most of these are run by the Moffitt Cancer Center, although there are a couple of exceptions there which I've marked in red. And you'll see that most of these trials are actually pretty small. So the N equals, that's the number of patients in the trial. You can see generally it's between about, what is that? Between about 14 and 34. So these are small trials, just trying out approaches. And the idea is that if these are successful, then they will progress to larger trials. But there are two exceptions there, which are the ones not run by Moffitt. There's a larger trial in London, and there is a particularly large trial taking place in Australia and the Netherlands, which is a randomized controlled trial. So that's the most ambitious of these ones, the one with the biggest resources, but also the one that will take longest to run. And you can see here that there are mainly two strategies here. One is called multi-strike or extinction therapy that I'll be talking about, and the other one is adaptive therapy. And they're being trialed in various different kinds of cancer. So there are many in prostate cancer, but also here breast cancer, melanoma, and other cancers. So what we're trying to really understand is, can we use mathematical modeling and experiments to predict the outcomes of these trials and to help design better trials in future? So this is the first strategy. If we can't cure, then let's aim to maximize survival and also quality of life. This approach goes by different names. It's largely referred to as adaptive therapy, which that name comes from the way the strategy is administered, in that the clinician monitors how the tumor is responding to the therapy and adapts the dose or the intervals between doses in response. Is also known as tumor containment, and that focuses more on the mechanism by which this therapy works. So these names are somewhat interchangeable. They're not exactly the same, but they're used for broadly the same approach. This approach was first studied in depth in a couple of studies published around 2008, 2009. The second one there, you see Bob Gatenby was the first author, and this paper has um, garnered many, many citations and a lot of attention. The first of those papers, published by a couple of mathematicians, was largely overlooked for a very long time because those mathematicians didn't really follow up on it. They said, here's a nice model, it shows that this works. They went on to work on other things. And it was Gatenby really who took this idea and ran with it. But you can see there from the descriptions that they're essentially describing the same kind of idea, that we can use lower doses, reduced chemotherapy, to improve survival times due to the effects of competition between resistant and sensitive tumor cells. That's the basic idea. So you suppress the resistant populations by maintaining sensitive cells. This is this figure here shows results of the first clinical trial of this approach, which was published a few years ago. And what this shows in the survival plot on the left-hand side is that the survival of the cohort that used this new approach, which is in red, that survival curve comes down much more slowly than the survival curve of a contemporaneous cohort that used a more conventional treatment strategy in blue. There are some caveats with this trial. So this was not a randomized controlled trial. This was a trial that considered a cohort where they applied their strategy and compared it to a contemporaneous cohort that received different strategy. That were not themselves within the trial. So that is a caveat. But still, these results are pretty striking. And I attended a conference in September where they reported that one or two of these patients are actually still alive on this protocol, which you can see that for the contemporaneous cohort, that would be really unheard of. So just to summarize what this approach aims to do, the goal of adaptive therapy is to enforce a stable tumor burden by permitting a significant population of chemosensitive cells to survive so that they in turn suppress proliferation of the less fit but chemo-resistant subpopulations. That's what Gatenby wrote when he first proposed this idea in 2009. So we can take from that there are several key elements to this hypothesis then. So we've got the idea that we can enforce a stable tumor burden. That's important. We need some way of doing that. We need the sensitive cells to suppress proliferation of the resistant cells. And there's an assumption here as well that the resistant cells are less fit. That means they have lower evolutionary fitness. They have lower net growth rates than the resistant cells in competition. So this paper by Gatenby then proposed this idea, and it then immediately suggested these questions. Does resistance really incur a cost, a fitness cost? Can resistance be suppressed? What's the mechanism by which we can suppress resistant cells using sensitive cells? And how much will this benefit patients? Even if this idea works qualitatively, how much benefit can we expect? Is it really worth trying? So my collaborators and I set out to answer these questions when I started my postdoc in Montpellier back in 2014. So I worked with the lab of Dan Fisher, and that included Katerina and Ahmed and Liliana working in that lab. And they were using at the time, a human colorectal tumor cell line that is well studied. And they selected some of these cells to be irreversibly resistant to a drug. And they did that by exposing the cells to the drug. Almost all the cells die, a very few of them survive. You then keep culturing those cells over many months under drug pressure. And eventually, they become irreversibly resistant to the drug. So they grow very happily with or without the drug. So what you see here are the growth curves just in monolayer culture, just in a dish. The blue is the original cell line, and the two red curves, one of them is the growth of the resistant cell line without the drug, and the other one is the growth of the resistant cell with the drug. Now, I forget which color's which, but it doesn't make much difference. What you see is there's basically no difference. These cells are perfectly happy to grow with the drug. Whereas if you put the wild type with the drug, in green at the bottom here, it quickly dies. So this gives us a nice experimental system. What you see, though, is that these resistant cells are indeed paying a fitness penalty. They're growing more slowly in isolation than the wild-type cells. They also tried these cell lines in nude mice, and they saw a similar result, that they grow more slowly in mice when they're resistant. So yes, at least in this cell line, we had a resistance cost, and that was encouraging for running these experiments because the suggestion was that a resistant cost would make this approach more effective. So the next question is, can resistance be suppressed? We need somehow the sensitive cells to inhibit the growth of the resistant cells. How can we do that? So here the lab turned to tumor spheroids. Tumor spheroids are clumps of tumor cells that grow in suspension in a liquid. They're very small, but they recapitulate many of the features of very small solid tumors. So this is going from cells moving around in a dish to having cells in a clump where they grow outwards as a ball of cells. And this recapitulates particularly the oxygen gradient. So oxygen has to enter the spheroid from the surface, and this sets up an oxygen gradient, which means that the cells near the middle have very little oxygen. And here we see necrosis. We see cells that are starting to die due to lack of oxygen. We also see a distribution of glucose, which comes from the medium that's outside. And this means that cells proliferate more near the edge of the spheroid than they do internally, where we get, as I say, necrosis. And this kind of system is very well understood. It's been studied in detail because it's a very controllable system that can be studied in the lab. So this gave us a way of looking at how this system might work under very controlled conditions. So from this understanding of spheroids, I put together this simplified diagram to design a model. So what we know is that cell proliferation is inhibited by a cost of resistance, by distance from the edge of the spheroid. So the further away you are from the edge, the less often you proliferate. And one reason for that is simply mechanical forces. The cells are squashed when they're deep inside the spheroid. Related to that, hypoxia, when the cells are deprived of oxygen, they proliferate more slowly, and that mechanism is well understood. And the drug, of course, the drug inhibits cell proliferation of the drug-sensitive cells. Hypoxia and the drug also cause cell death. So the nice thing is we could quantify these things. So this curve here shows how division rate varies with oxygen concentration. This is based on previous literature, the nature of this curve, and I built into this the cost of resistance, assuming that the cost of resistance acted across all the oxygen concentrations. In terms of competition for space and how cells would be inhibited by being far from the boundary of the tumor spheroid, I put together a agent-based model, which means that I have a grid of spaces that cells can occupy. And in order to divide, a cell needs to have space. If it has space immediately next to it, it can simply divide into that space. If not, it has to budge other cells out of the way. So the way this works is that the cell finds its shortest route to the boundary, and then it pushes cells in a straight line to make space for it to divide. But if it's too far from the boundary, then it can't do this. And I can change that parameter to control the extent to which physical constraints inhibit proliferation. The treatment effect, that was something we could quantify. What I found is that the effect on proliferation rate was approximately linear, as you see here. So I could build that into the model. And beyond a certain dose, we see the treatment also has an effect on death as well. So I built all of this into a model. As I say, I designed a fairly simple model, a two-dimensional cellular automaton. And here, this is first growing a simulated tumor spheroid in the absence of any drug. So the green here is an oxygen field. The more green there is, the more oxygen. That's coming from the medium surrounding the spheroid. The blue are the sensitive cells, those that are sensitive to the drug, and the red dots you might just about be able to see, which are blown up here. These are the resistant cells. And the darker the cells are, the more hypoxic they are. That is, the less oxygen they have. So this is the initial conditions. I've started here with a couple of resistant cells that are randomly located within the simulated tumor spheroid. And then what happens is it starts growing. Uh, and, oh, I'm sorry. There we go. Let's see if we can get this one growing. Okay, there we go. So this is growing in the absence of a drug. What we see is the spheroid just grows outwards. Because the resistant cells pay a fitness penalty, they divide slightly more slowly. That means they tend to get left behind. So even if they're initially at the edge of the growing tumor, because they're dividing more slowly, they get overtaken and they end up pushed into the hypoxic core, and therefore they can end up being eliminated in this particular scenario. Now, what if we grow with a drug? So this is with a high dose of drug. The inhibitor here, the drug is shown in orange. So you will see that in this case, the tumor spheroid starts to grow, and then the inhibitor is introduced, and that will kill off all the sensitive cells. There we go. And then the resistant cells reform a spheroid and they grow outwards. Now, neither of these outcomes is very good because in the first place, you end up with a very large drug-sensitive tumor. In the second case, a very large drug-resistant tumor. So we want something perhaps intermediate to see if that will successfully contain the resistance and also uh, result in a smaller tumor. So in this case, in the lower grid, what we see is a lower dose, and this dose suffices to arrest the growth of the sensitive cells, the blue ones, but without killing them at an appreciable rate. So in this case, what you see is when we introduce the drug at dose 20, it essentially more or less freezes the tumor. And that means that the red cells, the drug-resistant cells, end up trapped inside the tumor. So you're not trying to eliminate it, you're trying to simply constrain the tumor and maintain enough sensitive cells to trap the resistant cells. And so this shows how the idea can work in a computational model. And in summary, what we have here is on the left-hand side, we see different outcomes for 5 doses, going from no dose, a very low dose, which is too small, 10, which is still too small, 20, which is the containment dose I just showed you, and 50, which is too high because it kills off the sensitive cells. And then you see going down from day 7 up to day 28 how the simulated tumor spheroid progresses. And the best outcome here is at dose 20, where the spheroid remains small. What you see at the bottom is here I've just highlighted the sensitive and resistant cells. And what you see is that in the containment case, most of the cells at the end are still sensitive to the drug, as I just showed you. So in terms of growth curves, you get what's shown on the right, where if the dose is too low, then the spheroids simply take off, as in the red and the blue. If the dose is too high, initially you get a good response, but then the tumor bounces back. You get relapse due to resistance, and so your spheroid ends up big. And if you apply the containment dose, you end up on average with a smaller tumor. Now, what's shown here is a median case, and you can see that in fact there is quite a lot of variation either side of that median and mean. So the median is the solid line here, and the mean is the dashed line, and then you can see a lot of variation within this credible interval here. This just shows you the kind of variation I saw in simulations. And so I'll come back to why there was such variation. And this is how it compared with the spheroids. So we were pretty happy with this, that the computational model, which has all those transparent assumptions based on the experimental results and previous literature and so on, was in good agreement with what we saw in the actual spheroids shown here in the right-hand half of the slide. You'll notice that the growth curves, I've cut them off at around spheroid size 0.2 millimeters cubed. And this is because the spheroids don't grow well beyond that size. They tend to just level off. So you wouldn't expect agreement between the experiments and the simulations beyond that size because basically all these curves then just level off and you get nothing interesting anymore. Now, is this surprising? I don't think this is actually a very surprising result because, as I say, tumor spheroids are so well understood. I could set up a model that built in all the important mechanisms and parameters of these spheroids, and so we would expect that it would be in good agreement with the experimental results. But still, it's nice to see it come about like that. Now, I said that there's still a lot of variation in the results, and it turns out that the most important parameter is where the resistance emerges. Because if the resistant cells happen to be initially near the boundary of the tumor, then they won't be as effectively contained. And so what I've plotted here is on the x-axis, this is the initial distance between the outermost resistant cell and the periphery of the tumor spheroid in terms of cell diameters. So 0 here corresponds to the cell being on the edge of the tumor spheroid. 1, it's a cell diameter away, 2 cell diameters away, and so on. And on the y-axis is the proportion of simulations that had more than 10% resistance after 28 days of simulation. And what you see is that this is extremely dependent on where the resistant cells were initially located. So if you initially happen to have resistant cells at the boundary of the tumor spheroid, then there's a high chance that some of those resistant cells will remain at day 28. So this was based on the model that I just showed you. What I can do, of course, in the simulation is just turn off that restriction on crowding. So I can say that wherever a cell is within the tumor spheroid, it can push other cells out of the way to divide. So I just make that radius unlimited. And what you see then is that many more of these simulations have resistance remaining at day 28. Even if the resistant cells were initially 2 or 3 diameters away from the periphery, that's sufficient for most of the simulations to still have surviving resistant cells. So this confirms that at least within the computational model, this crowding inhibition, so the spatial constraint, is what makes this approach work. It's the fact that these cells are physically inhibiting each other from dividing that is the key mechanism, at least within the simulations. And the nice thing about simulations is you can alter parameters like that you wouldn't be able to alter in the experimental system. So in answer to our questions then, in this initial study, what did we show? Resistance does incur a cost, at least in this cell line, for this particular drug. Resistance can be suppressed. So the question then was, how much will this benefit patients? And here we turn to a different kind of model, and I teamed up with another mathematician. So Yannick Vieussat is now a long-term collaborator of mine. We've been working on these models for several years now, and our first project was to come up with a nice little paper on this kind of system, and it turned into a huge project and a very big paper. So what we started with was a very general model. We've got 2 types of cells, sensitive and resistant, S and R. And each of these has a growth rate, G, so GS and GR, and the growth rate can depend on the number of sensitive and resistant cells. And in the case of the sensitive cells, it can also depend on the drug dose. So that's a very general model. We don't assume any particular form for this growth rate. So we don't say what mathematical expression we insert there. We just say that these G functions have to have certain properties. We assume that GS is non-increasing in C. So in other words, when you increase the dose, the growth rate of the sensitive cells doesn't get bigger, which basically means the drug works. That's a pretty justifiable assumption. And the second one is that GR, the growth rate of resistant cells, is non-increasing in S. That is, there's some kind of competition. So the sensitive cells can inhibit the resistant cells. The more sensitive cells you have, the slower the growth rate of, or the lower the net growth rate of the resistant cells. Those are the basic assumptions we made. And we, or rather Yannick, formally proved that these results hold very generally for prolonging survival. So under those very general assumptions, then the optimal strategy is to maintain the tumor burden as high as possible, so long as some sensitive cells remain. So the idea there is if the only way that you have to suppress the growth of the resistant cells is by maintaining sensitive cells, then you want to maintain as many sensitive cells as you possibly can. So ideally, what you do is you allow the tumor to reach as large a size as is acceptable, and then you basically freeze it there, and that's how you can maximally contain resistant cells. More practically, you use this approach called adaptive therapy, which means you modulate the dose or the gaps between doses so that the size remains approximately constant. And the worst option is to maximize the kill rate. That is the maximum tolerated dose to apply as much drug as the patient can withstand, which is the current strategy. But we also investigated how the quantitative results depended on various parameters. And we found the key thing is the intensity of competition between sensitive and resistant cells. So on the left here, you see growth curves over a certain range where I've parameterized these three different models, von Bertalanffy, Gompertzian, and logistic. These are alternative models of tumor growth with different kinds of competition, as shown at the bottom here. I parameterized them all to have similar shapes so that if you had some data, you might not be able to tell which of these curves was a good fit to your data. And yet these three models have very different predictions about the relative benefit of this approach relative to the conventional way of treating. So what you see is that if you used a von Bertalanffy model, you'd predict huge benefits from this approach. Gompertzian, reasonable benefits, and logistic, very weak benefits. And we really don't know which of these is an appropriate model. So this is a big caveat. We also looked at when containment might be suboptimal. And so we found that this would be the case firstly, if you violated the assumptions. So if all cells are sufficiently sensitive to treatment, then, the model doesn't really apply. You might as well just apply high doses if you can also contain the resistant cells using the drug. So in other words, the resistant cells aren't fully resistant. Also, if we assume that sensitive cells don't inhibit the growth of resistant cells, then obviously this approach won't work either. There are some factors excluded from this basic model. So we don't include any kind of mutations or plastic responses induced by the treatment. So if the treatment itself makes cells more likely to be resistant, it induces cells to become resistant, then that could change the results. We did look at just spontaneous random mutations, and we found that that doesn't matter very much. Also, importantly, the risks from sustaining a high tumor burden might outweigh the benefits of containing resistance, right? Having a very large tumor inside a patient is not generally a good thing. There are risks from metastasis and from cachexia and so on. So that has to be balanced, and that's not included in the model. Although these are things that Yannick and I are continuing to investigate. So I said that that's the work we did previously. So one of the reasons I recently went back to Montpellier was to catch up with Dan Fisher's lab. They've been working on new preclinical models, in fact, a couple of different models, but I'm going to focus on one in particular here, just in the interest of time, led by Alex DeLupi here. What he's been doing, he started out with two-dimensional competition assays, the same as before, where you put these cells together in a dish. And what he's been running is many of these assays. He then applies a cell sorting procedure where each of these dots here is a cell. You figure out this way whether a cell is sensitive or resistant, depending on where it's located in this plot. Based on that, you calculate the frequencies of drug-sensitive and resistant cells over time during these pairwise competitions. If you then transform these curves according to a particular transformation called the logit transformation, the log of the frequency ratio, then they look like this under different drug doses. If you take the gradient of these curves, it turns out that that gives you the selection coefficient. So that tells you the fitness penalty paid by the resistant cells versus the sensitive cells by looking at the gradient of these curves. So this is the procedure we followed in analyzing these results. In this case, this is a lung cancer cell line, and Alex has been using targeted cancer therapies that are used in the clinic to treat lung cancer, so very clinically relevant drugs. And what he found out, he tried this experiment with many different initial frequencies of resistance. He tried it in high density and low density conditions, and consistently what he found is that when these cells are cultured under drug pressure, then the fitness of the resistant cells decreases when these resistant cells are more dominant. So in other words, resistant cells are fitter when they're rare. They like to be surrounded by other sensitive cells. The reason why we think this is because the resistant cells are paying some kind of fitness penalty for being resistant. Maybe they produce more waste factors that harm other cells. Maybe they consume more of the growth factors in the culture. And so these are not very nice cells to be around, and the sensitive cells are much more pleasant to be around. So the resistant cells prefer to be surrounded by drug-sensitive cells. The dashed line there is what you would predict if you just looked at the growth rates of the two types in isolation. So that is actually not a good prediction, even for the control, the untreated cell line. But you can see roughly that what you'd expect that to be, this is essentially an extra dot here when the initial frequency of resistance is equal to 0, then is equal to 1 or 0. This is when you'd see the that's when you'd see a monoculture of either resistant or sensitive cells. That those would be the endpoints of this line. But you see that doesn't give you a good prediction for when they're in competition. So Alex then, again, he proceeded to tumor spheroids, and in Montpellier, they now have these beautiful confocal microscopes, which allow them to get these extremely detailed images of the spheroids, showing the individual cells, and they can do that as a stack all the way through the spheroids. So they have perfect information, which we didn't have before. Here we have the sensitive cells in green and the resistant in orange. So you can see also where they're spatially located. And you can then extract the resistant cells. You can cluster them. You can identify how many clusters you have, where they are, and so on, which is amazing information. And I have a PhD student who's now using this information to produce more sophisticated spatial models and machine learning methods and so on, which isn't quite ready to present yet, but is very exciting. So Alex was able to then compete these cells either in initially 10% resistant, as you see here, or up to 50% resistant. And what you see in both these cases is that the resistant cells tend to end up not very close to the periphery, further from the periphery than you might expect by chance. And that also holds here in the 50% resistant case, that the resistant cells tend to be located somewhat away from the boundary, which is what you'd expect if they're paying a fitness penalty. So again, Alex did competitions, and what he found in this case was that in the absence of treatment, there is the opposite effect, that resistant cells are fitter when they're dominant. So here we have increasing fitness with increasing initial frequency of resistance. And this makes sense because if what is suppressing the resistant cells in these spheroids is mostly this competition for space. Then if they're surrounded by their own type, they're going to do better than if they're surrounded by the fitter sensitive cells, which are going to tend to crowd them out. So we think that the crowding effect dominates in 3D, whereas the effects of these paracrine factors, whatever's being produced in the medium, that dominates in 2 dimensions. So Alex has moved on to a preclinical trial, putting the same cell lines in nude mice with the drug. He's demonstrated that that without treatment, the resistance is costly in the mice. And we expect, so we see a large fitness cost in vivo, and we expect that we might see a positive correlation here between fitness and frequency, though we have yet to find convincing evidence of that. So the drug does shrink these mouse tumors, making it a good model. And what I'd love to be able to show you is adaptive therapy versus maximum tolerated dose, actually trying out this strategy in mice with labeled resistant cells. It's not quite ready yet. So they have some preliminary results, but anything in mice is complicated. And so I'd rather not present results from that yet, but it's something I'm looking forward to being able to present in the near future. So I'm going to talk briefly now about the second strategy. This is a shorter part of the talk. The idea here is that if we can cure a tumor, if we know historically that a drug does sometimes cure a tumor, then let's try to maximize that cure probability rather than go for containment. So this is an approach that Gate and B and Co have called extinction therapy. We also call it multi-strike therapy. So the idea is that you apply an initial therapy to reduce the population size, that's to shrink the tumor. And that not only shrinks the size of the tumor, but it also decreases the diversity of the tumor. So you've got fewer types present, and each of any rare type is going to be very rare. And then when the tumor is at a small size, you apply another treatment or a series of treatments to kick the tumor while it's at its weakest. And what you hope to do is to exploit stochastic extinction and to drive the tumor to extinction that way. So this is the basic concept. We have an initial tumor shown in black here. The tumor declines under the initial therapy, but what typically then happens is that the tumor relapses because resistant cells, some rare resistant cells, as in the previous experiments, rescue the population and bring it back. So you started out with a mostly drug-sensitive tumor, you end up with a resistant one. What doctors typically do is then they wait till the tumor comes back. They say, it's come back. And then at this point, when the tumor is regrown, they try a different treatment. And often the cycle then repeats. Now, the concept here is maybe it would be better to treat at some other point rather than waiting for the tumor to regrow. Might it be better to treat before to switch to a second treatment before the tumor reaches its smallest size and min, or perhaps close to that smallest size or shortly afterwards? That's the question here. So to answer this, I teamed up with a brilliant master's student, Srishti Patil, and an undergraduate from Johns Hopkins, Arman Ahmed, who's now applying for PhDs. And one thing that Srishti developed and then Arman also used was stochastic simulations. This means we simulate the system. You can see here we have exactly this. The tumor decreases under treatment. It then starts to bounce back. We then apply a second treatment here. Sometimes the tumor goes stochastically extinct. This curve keeps going down. And sometimes it gets rescued by cells that are resistant also to the second treatment, and we get relapse, which is called evolutionary rescue in the evolutionary theory literature. And what we see is that if we switch when the tumor is small in these simulations, we get a higher probability of extinction, PE, than we do if we switch too late. So it's best to treat when the tumor is, to switch when the tumor is smaller, where this red line. This red line's lower than that red line. That's stochastic simulations, sure, but we also wanted to get some analytical mathematical results. So Srishti developed this model, mathematical model. What she assumed is that we start with a tumor at the end of the gray period here. We apply a treatment, and during that treatment, we apply a differential equation model, which means it's a deterministic model because we expect very little variation while that first treatment is applied. At the end of that first treatment, we then switch to a second treatment, um, and that begins our second period E2, our second environment E2. And at that point of switching, we look at how many cells are resistant to the second treatment at that point. And based on that, we calculate what will be the probability that the second treatment will drive the population to extinction. So in mathematical terms, that means using a differential equation model to model the growth of these different populations. So this is sensitive, resistant to the first treatment, resistant to the second treatment, resistant to both treatments. This is the growth terms during that first treatment. And then we've got mutation terms. So these cells can mutate. So we have transitions between the different types. Then what we do is we calculate the probability of extinction, PE, based on the probability of extinction due to the resistant cells that already exist at the time we switch, multiplied by the probability of extinction due to mutations that occurred during the second treatment. And mathematically, that has a form as shown in this slide here. Sorry, there we go. So this gave us a very nice way of obtaining analytical results as well as the simulation results. So we have an analytical solution of an approximate model. We have the extinction probabilities, we have the agent-based modeling, and then we did a great deal of sensitivity analysis and looking at the robustness of these results. And what we found basically is the best time to switch is when the tumor is very close to its smallest size. So on the x-axis here, you have the size of the tumor at the time we switch from the first to the second treatment. And the dashed curve here is the smallest size that it attains. And on the y-axis is the extinction probability. Now you can switch either before or after that nadir, the smallest size. And what we see generally is that if you switch a bit after, that's better than switching a bit before. But the best of all is to switch pretty much exactly when the tumor is at its smallest size. Nevertheless, it is better to treat a bit too late than too early, unless you assume that there are no resistant cells present at the start of treatment, in which case it doesn't make a lot of difference. So we found some further results in that study as well, which are summarized in which you can read more about in the paper. We found that higher treatment efficacy is not always beneficial, which is interesting. Cost of resistance is not necessarily beneficial. Same thing actually holds for adaptive therapy. Extinction probability is insensitive to the carrying capacity, that is how big the tumor can grow. But it does depend on the death rate and the turnover of the cells. Most importantly, though, this two-strike therapy is only going to work in small tumors. That's our prediction. So to get this to work in practice, you will probably need to apply more than two strikes. And we predict that if you do that, it might actually be able to eliminate reasonably large human tumors. So back to the main messages, as I summarized at the beginning, really, if we can't cure, then perhaps we should aim to contain tumors and maximize survival. But if we can cure, then we can use evolutionary theory and mathematical modeling to predict the best time to switch treatments. So with that, I'd just like to thank all the people who were involved in this research. And yeah, to open up for questions, and sorry for overrunning a little bit.
Thank you very much, Rob. That was great. Marie, do you want to take it over and see who has questions?
Sure. You can also send some question to the chat if it's not easy for you to talk, but otherwise just step in and ask.
And also, as I said, sorry, if you can say very quickly what your domain of expertise is before asking your question, I think it can be useful for Rob to know if you're a philosopher, medical doctor, or biologist, for example. I have a question. If I have many questions, it was really, really useful and quite fascinating. You mentioned several times metastasis, but one thing that strikes me is that in the field, it seems that we're increasingly going towards the view that metastases are in fact very early and often micrometastasis. And I was thinking that that might have very strong consequences potentially on what we should do and when. So it's a very general question, but the question I expected to be a little bit mentioned more in your talk. So if you could say a little bit more about this, that would be very, very interesting. Oh, sorry, I forgot to say I'm Thomas. I'm a philosopher. Sorry.
Sure. Yeah, that's a great question. The models I presented are they're designed to be very simple in general. So we're trying to get to the general concepts and mechanisms by which this approach could work. And therefore, we assume we just have one population and we don't distinguish between multiple metastases. Other people, such as Jill Gallagher at the Moffitt Cancer Center, have investigated models that are specifically based on multiple metastases in the case of adaptive therapy. So I think that is very important because many of the clinical trials are being run in metastatic cancer. So it really does matter how, particularly when we come to the questions of how large are these populations. If you have many small metastases, as you say, maybe we we can model this with very small population sizes. And that could also be interesting for this multi-strike therapy. That if we, and I think we comment on that in the paper, that if we have many small metastases, maybe that this approach could work. If you can remove the primary tumor, so you've only got small residual tumors, then perhaps this could work. It might be more appropriate then. Um, yeah, so I think that that is a very important. One question there is just how we parameterize a model for metastatic cancer. How do we set it up to be different? What is it that would be different from this simpler model here?
Thank you very much.
Caroline?
Yes, can you hear me?
Mhm.
Yeah, so hi, I'm Fridolin. I'm a colleague of Thomas and Marie's, and I'm a philosopher and a computational biologist by training. Um, so in the first part of your talk, it became clear that interaction between cells is very important for the success of these adaptive therapies. So have people tried somehow to intervene on not directly killing or inhibiting cell growth, but on processes related to cell interaction?
That's an interesting question. You mean to enhance the strength of the competition between cells?
For example, yeah, or in general, just processes relate to how I don't know, cell adhesion or whatever, I don't know.
Not as far as I know, but that could be an interesting avenue. Yeah, to perhaps enhance if you had, I guess, one drug that constrained the tumor and another that somehow enhanced the competition, that would be
Yes,
intriguing.
yes.
I'm not aware of a way of doing that, but yeah, that's an avenue I've not heard discussed. Um, I'm very interested in how cells do interact, and this is another thread of my research. But I've not combined that with thinking about how we could enhance that. Well, one thing I have thought about is how different tumors might have different tumor types might have different kinds of interactions between cells, different kinds of spatial structures, and that might make these therapies more or less appropriate. Because I think there's very little understanding at the moment of which cancer types this approach might work in from a basic biological point of view. Uh, a lot of the rationale for these trials seem to be based on what the drug, whether we have an effective drug and whether we have a ready cohort and whether there's a, clinical rationale rather than what's happening to the cells, how are the cells interacting. So I think that really is an ambition of our research to be able to predict what features you'd look for in tumors and then therefore be able to predict which tumor types might be good targets.
Okay, thank you.
Marie, I think that Andreas wanted to ask a question.
Yeah.
Yeah, so hi, I have a question also. So I'm I'm an MD PhD biomedical scientist and recently also a philosopher. So I have three theses. So the thing is, so the question is, so the central question is in fact you have these two, you have the sensitive cell types and the resistance one. And there is Phil Jones, recent from Cambridge, who is system on esophageal cancers. He has two cell competitive, and he says also when you erase the competitive cell types, then the tumor will grow more. But when you have the competitive tumor cell types, then you have an inhibition. So I think this is a little bit related to your observations that you have. So I don't know if you know his so his name is Phil Jones.
I certainly know of him. Yeah, I have met him once or twice. I'm sorry, the signal wasn't perfect. So do you mind repeating the question?
So yes, there are two in the Phil Jones theory. There are two types of tumor cells. There are the competitive tumor cells and the non-competitive ones. So the resistant ones, they have driver mutations. And if you eliminate too much the competitive ones, then you get more of the resistant cell types that are in fact, and the system that he has is resembling a little bit what you have with the sensitive cell types and the resistant cell types. So that would be one way to. So I don't know if you have discussed with him this
I've
point.
I've not discussed with him that study. I'm more aware of his work on the skin and maintenance of homeostasis in the skin and so on. But yeah, yeah, if you could send me a reference to that paper, I'd be very happy to
So it's
read
hard
more
to.
about that research. Yeah.
Okay, just one other question. So there is, of course, the tumor. So your model is highly reductionistic. You have only, so you are focusing, in fact, on two cell types and then treatment. But when you have the tumor, you have the microenvironment is huge, and there are a lot of interactions going on with immune cells, blood vessels, matrix, so and so forth. And to model all this is very complicated, highly complicated to do that. So, I
Yes.
mean, if you are so in your system, it's hyper reductionistic, and the thing is that so I don't know if you can explain everything with this simple way to do that
Absolutely,
because
we
the
cannot know. So the idea of the model the modeling that I presented today was really to test conceptual models. So people have proposed these hypotheses, maybe this could work. And so what I set out to do with the lab and with Yannick was to see if those ideas could work at all. Is there any theoretical basis? People often have mental models of how things work. And what we found is that some of the mental models that people had written about were incorrect. So, for example, people had this mental model that if we turned a treatment on or off, then when the treatment was on, the sensitive cells would go down and the resistant cells would go up, sure. But then they thought that when they turned the treatment off, the sensitive cells would go up and the resistant cells would go down. And there's no model we could come up with where the resistant cells would go down during those treatment intervals. And another case is the idea of the cost of resistance. Gatenby originally said that this was a very important assumption of his approach, that there must be a cost of resistance paid by resistant cells. And what we found is that that is not essential. It's useful, but it's not essential. So the idea of our modeling was really to sharpen those hypotheses and to test whether these ideas actually could work, to give us a more rational basis. In terms of predicting outcomes, I think what we found is that there are just so many open questions at the moment. We are wary to make very precise quantitative predictions. As I showed you, it depends which model you choose for starters. And then it would be different if you started to, as you say, include microenvironmental effects, the immune system, many other factors. The problem there is always parameterizing, and that's why we team up with experimental biologists. And so what the experiments that are ongoing now in the mice should give us what we need to start parameterizing this. But still, it's nude mice, right? We don't even have fully functioning immune systems, and we're moving on to immune competent mice. But it's in order to parameterize models in a human is another step. So we're going there step by step to understand all these different mechanisms, all the parameters you need to have. And when you get data from clinical trials, unfortunately, you can fit a million different models to the kind of data that come out. So there are people who are attempting to model the clinical trial data, but we're also trying to just sharpen those hypotheses and sharpen the questions. And I fully concede that it's far more complicated.
Anybody else? Thomas.
Unless someone else has a question. Okay, so first I wanted to apologize because I see that some people just entered the room, and I'm so sorry. I gave, it seems, 2 times, yeah, I'm so sorry. It was 4 p.m. French time, and I'm so sorry I made this confusion. The real question was, I really like.
Okay, so like let's try one data.
One practical and sort of historical question I had for you, Rob, was I really liked what you just said about the utility of modeling often can be to sharpen our hypothesis and, for example, eliminate assumptions or make better assumptions. I think this is extremely useful. One historical question I have for you is, to what extent Gatenby himself is mathematical? This is something I don't know well. So did you bring the mathematics to something that was initially relatively conceptual and theoretical, but with very little mathematics? Or was it modeling based and mathematic based from the start, and you're just sort of improving that? Yeah, this is really a question of curiosity here.
In the supplementary material of our 2021 paper, we review all the previous mathematical modeling that had been done to that point. In brief, the best mathematical modeling papers at that point there was one by Martin et al. From the early 1990s, I believe, which was of a similar idea. And then there was this paper by Monroe and Gaffney, and those are the two papers that we mostly built upon. Because they formulated this problem in terms of these differential equation models, which are standard and tractable and very flexible and so on. Gatenby's 2009 paper, he teamed up with a mathematician, but I've never been, for the life of me, able to understand that model. And I don't know anyone else who does either. So they did have a model, but that model didn't really lead anywhere. I think because it was a very non-standard kind of model. But what Yannick and I wanted to do, by the time we were doing this work a few years after that original 2009 paper, there had been a few studies, but they'd picked particular differential equation systems or particular agent-based models. They all seemed to show that this approach could work, but we had no way of comparing those different approaches or seeing what it was that actually made it work. And so our idea was to go for this very, very general approach. That's why we wrote these general functions, and then to establish what properties those functions need to have. And then as long as you formulate any model that has those properties, which all the pre-existing models had, then we could predict that, yeah, qualitatively, you're gonna find adaptive therapy works. So that then none of those previous results were surprising anymore. They were all just variants on a theme, and we knew what parts of them made it work. And then everything, all any other assumptions they put in, like cost of resistance, like particular forms of spatial structure, whatever, were just, they might change things quantitatively, but they weren't going to change the outcome. We could tell you what the outcome would be just by looking at the properties of the model itself. So we have tried to do something similar with other approaches to kind of generalize the methods. So yeah, we were building on prior work, but we tried to generalize it and to synthesize it all so that we could compare everything that had been done before and put it within a single framework and say, these are all just variations on a common theme. And this also helped us understand what else could be done. What's missing from these models, what might change the outcomes.
Very interesting. Thank you.
If nobody else has a question, maybe I can ask you, Rob, could you please tell us a bit more?
So
you were
maybe
saying,
ask another say.
yeah, you can go ahead. Yeah,
Marie,
please.
I think
Okay, I can go ahead.
Marie, you
So
didn't ask a question. It's probably better that you ask a question. I think it's fairer.
Okay, it doesn't matter. Yeah, so you were saying that you're going to try to expand your model to also include the immune systems, like more complicated parameters. How are you going to? Oh, yeah, and you were saying it's difficult to parameterize such models, and you need a lot of data to do so. So I was wondering if you could tell us a bit more about how you're going to do this, especially because when you have a lot of data, then sometimes you're just flooded with data and you can just overfit your model, right? So you can always find a model that will fit to the data, but how will you make a model that makes sense conceptually, basically?
So my former PhD student, Blair here, who's the one with the tattoos at the front, who recently successfully defended his PhD, there is a chapter in his thesis where he introduces some immune factors into these kind of models. So he has some preliminary predictions there. The other thing Blair developed is a very nice way of fitting these models to data using MCMC so that we get nice Bayesian fits, as we did in our previous collaboration, Mary. And that's being taken forward by my current student, Kate Bostock, who is applying those methods to the new data. So in terms of avoiding overparameterization, I think these Bayesian approaches are nice because if you have too many parameters, instead of overfitting, what they tell you is they give you very broad distributions. They say we have a huge amount of uncertainty about those parameter values, and that means you can you don't overinterpret your model. So Kate and Blair have developed these ways of fitting to the lab data and getting some nice inferences from there. So essentially repeating the analysis of Alex that I showed you, but with a Bayesian approach, so we get all the uncertainty as well. And Kate is further developing methods for using AI approaches to take those images. And where we have sequences of images, she's developing a method that will be able to enable us to predict what happens next. And to try to extract information from those images so that we can do the same kind of modeling, which you cannot do with these conventional approaches. But with image processing methods, she has a proof of concept that we can start doing that kind of thing. Again, she puts in some constraints there. So she puts in some constraints on how the cells can grow and so on. So it's a mixture of using AI methods and the more conventional mathematical modeling. So I think that's a nice way forward. In terms of, yeah, modeling the immune system, as I say, Blair has done some initial work there. But I agree, it is hard to parameterize. I'm always a bit scared of modeling the immune system for that reason. But I think we can make some progress if we keep things simple and we again test hypotheses. So we ask experimentalists and biologists who know far more about those systems than we do what their hypotheses are and how they would design a model, and then we can see whether again their mental models for how this might work would actually play out.
Andreas, do you want to ask your question now?
If just to, okay. So just a question, because the problem with the tumor is in fact two-factor growth, and then you have invasion. So you have two types of
It's very difficult to hear
behavior.
you.
You have
Yeah,
the more this you
We
see
cannot really hear
extremely
you.
well. So do you hear me or not?
it's a poor connection, I'm
It's,
afraid.
Yeah, it's
so
very
do you hear me?
difficult. Maybe if you turn off your camera,
It's
we would.
difficult. So
Or you can also maybe type in your question in
no,
the chat.
I'm extremely close here. So is it okay now?
It's more a question of an internet connection than
Yeah.
being close to the
In the chat,
microphone.
okay. So there are
So I guess Marie-Andreas is typing his question. So let's see if someone else maybe has a question or comment while Andreas is typing. I have a very naive question again, but that would be my third question. So Marie, I understand if you prefer that someone else asks a question.
No, go ahead.
No, it's very naive. It's just that I was thinking that you were saying yourself that randomized clinical trial would be the thing, and you mentioned I think only one, which is a randomized clinical trial. Can you maybe say more on that trial and on why there are not more of them out there?
I think it's in terms of why, I think it's a compromise always between the expense and the amount of time it takes to run a full trial versus wanting to get quick results to move forward and test out lots of different ideas. So I think Moffat has wanted to kind of prototype lots of different approaches and see what works. I think the two approaches are complementary. But I think the European trial is the one that will eventually be more likely to win over clinicians because it is a controlled trial. It's a larger cohort. And I've spoken to some people who just said, yeah, this is the trial we wanted to see. that would actually convince us. But it is very tricky as well because so much will depend on the protocol and which cancer type they chose and which drug and so on. Yeah.
So you do not necessarily expect yourself to, in the end, consider that trial as the main trial that will either convince or not convince people. You're more like careful about what will be concluded from it, right?
It's always tricky with trials, isn't it? Because there's always the post hoc analysis. But yeah, we'd be looking at the primary outcome. So yeah, it will be uh it will be a very important result for sure. I mean, my collaborators and I, we've been quite careful not to be invested in one way or the other, right? We want to know when this might work, when it might not work. We don't have a horse in the race here. So we are always quite cautious to list all the cases where this might not work. And we are seeing experimentally in the mice, in fact, that sometimes it does not work. And so for us, that is equally convincing. We just want to know as soon as possible, one way or the other. My feeling is it's really going to depend quite a lot on the cancer type and the drug and the cohort and so on. That's why I'm a bit cautious in, even if that trial doesn't work, in writing off the entire approach, because there might be another situation where it's appropriate.
Does anybody else has a question?
Marie, I think Andreas wrote in the chat. I don't know if you can see, and maybe Rob as well.
I
So it's a
cannot
sort
see
of,
it.
it's a claim and then a question, it seems.
Okay, I
Do
don't
you
see
see
it
it?
yet.
Thomas, if you see it, can
Oh,
you
oh,
maybe
I'm
read it?
sorry. It was sent to me only. So let me
Okay.
do it for you, Andreas. Okay, so yeah. Andreas always needs my help. Let me see. Up. Meeting room. So that was the claim. And the question is, do you integrate these parameters? Do you try to integrate these parameters?
Sorry, which parameters?
I can read the question for you if you want. So it says there are two phenomena in tumor development: compaction and diffusion. The latter is related to invasion, which can be modeled. I am trying to model those in terms of causation. Not me, yeah? Andreas is doing it. The other is that models should integrate spatial transcriptomic, metabolomics, etc. Do you try to integrate these parameters?
Not yet, but yeah, I agree. We need better spatial data. So another thread of my research is looking at how different kinds of spatial structure within tumors result in different evolutionary dynamics, different degrees of heterogeneity, different frequency of resistance, different spatial locations of resistance, and so on. And that is crucial. And I think that's going to be really a crucial determinant of whether these approaches work. And if we can get that from spatial transcriptomics and so on, then that is going to be a game changer. I'm also interested in these uh, approaches. I mean, what we really need is three-dimensional structures of tumors, which have been very hard to intuit from two-dimensional slices from traditional H&E staining and so on. So tissue clearing approaches and things like this are very promising. And anything three-dimensional that allows us to understand the communities in which these cells exist, how they move between local communities, how they interact, the radii, the extent of interaction between cells, how they invade surrounding tissue, whether they disperse individually or in groups. All of those questions are fundamental, and I would love to be able to model those things more precisely. But often when I talk to people who collect that data, they say it is difficult to tell from static images, even the basic parameters such as what is the proliferation rate? because you can see how many cells are currently undergoing a certain stage of the cell cycle, translating that to a proliferation rate, translating a static image to a dispersal rate, a dispersal distance, and so on is very tricky. And I think that's where I would love to be able to parameterize models, because I can code those models. I have coded those models, but it's parameterizing them that is the real problem. And I think the experimental methods and the imaging methods are catching up. So I'm very excited about being able to do more with imaging data. And again, Kate is making great strides there using these machine learning methods to analyze and then fit models to images. And I think that is a great way forward.
That's a very nice way to end the seminar. Except if anybody has a very burning question, I suggest that you write to Rob if you want to continue this discussion. Rob, thank you so much. It was super interesting and also super nice to see you, even if via Zoom. I really hope we see you in Bordeaux soon this year, maybe. And I wish everyone a very nice evening.
That would be lovely. Yeah, thank you again. And I would be very happy to continue the conversation. If anyone wants to get in touch with me by email, then please do so.
Thank you.
Thank you very much, Rob. Bye.
Bye,
Bye.
everyone.